Today, I'm going to talk about Huntington's disease. My name's Roger Barker. I'm a consultant neurologist at the Addenbrooke's Hospital in Cambridge, the United Kingdom, as well as the Professor of Clinical Neuroscience. I look after patients with Huntington's disease, and we also do research both within the lab and in the clinic into this condition. In this topic, I'm going to take you a little bit through the background of Huntington's disease and then talk a little bit about certain aspects of it, with the hope that one will understand a bit more what this condition actually entails and some of the new developments which have happened more recently in terms of our understanding of both how it expresses itself in the clinic as well as some of the new treatments. So, Huntington's disease takes its name from George Huntington, who described the condition in 1872. It wasn't until 1983 that the genetic link to chromosome 4 was discovered, and in 1993 the gene itself was found, which codes for a Huntington protein. It's an autosomal dominant condition, and the abnormal gene codes for an abnormal form of the Huntington protein. Autosomal dominant clearly means that if you inherit one abnormal gene, then you will definitely get the condition. Now, for most people with Huntington's disease, they present between the ages of 30 and 50, but actually it can present at any age. There are people who present under the age of 21 who are said to have juvenile Huntington's disease, and there are people who have late onset Huntington's disease, the age of which is a little bit variable, but most people would say is over the age of 60 or 70. And certainly my experience has been in recent years, we're seeing more people presenting later in life with the first manifestations of this condition. Once it develops or becomes clinically manifest, it progresses over 20 years, and most patients will sadly die of this condition rather than with it. It's a relatively rare condition. In the United Kingdom, there are between 5,000 to 6,000 cases. In comparison, there are 130,000 cases of Parkinson's disease and over 500,000 people with Alzheimer's disease. So it's a rare condition. It is a genetic disorder for which we know the gene and we can test patients. So it does have special prominence in the world of research around neurodegeneration. Now, this is a condition which is in my part of the world. In Cambridge, there is a town up the A14 called Huntington. It is often referred to that by many physicians, which is incorrect because, as I say, it takes its name from George Huntington. We've also moved away from calling it Huntington's chorea, which was the movement or is the movement disorder associated with it, to emphasize that the disorder is much more disseminated in terms of its clinical features as well as its pathology. So we refer to it as Huntington's disease. As with all diseases, we often try and find someone famous who's had this condition. We slightly struggle with Huntington's disease. It is described in Ian McEwan's novel Saturday, where one of the main protagonists has this condition. But probably the most famous person is Woody Guthrie, that most people would never have heard of, who was a folk musician in the United States in the mid part of the last century. Actually, the Guthrie Foundation was set up in memory of him. That then led to the Hereditary Disease Foundation, and that in turn led to the discovery of the gene. So from Woody Guthrie, actually major research and discoveries have been made in this condition. So I think if we're going to start with Huntington's disease, it's always quite good to think, what do we actually understand about it sort of pathologically at the macroscopic level? What actually goes wrong in the brain with patients who have this condition? Well, the most striking abnormalities are atrophy and loss of cells within the brain. And exactly when that begins relative to when the disease presents, in other words, when the patient presents to their doctor with features of the disease, is unclear. But it is probably at least 5, 10, if not 15 years before the first obvious clinical manifestations. Now, the parts of the brain that take the major hit, that have the earliest pathology, are within the cortex, as well as within structures deep within the brain called the basal ganglia, where one slowly has atrophy of the striatal complex, namely the caudate and the putamen. And that particular pathology, the loss of the striatum, is actually still used to grade the pathological stage of Huntington's disease, with grade 4 showing the greatest loss in that structure and grade 0 to 1 showing the smallest amount of tissue loss and shrinkage. But as I say, Huntington's disease is not just confined to the basal ganglia, to the striatum, as some people used to think was the case many years ago, or at least 20, 30 years ago. It is a disease that affects all parts of the brain because obviously the gene is expressed in every cell, even though some parts of the brain take more of a hit than others. And this is illustrated in this particular slide, where we can see that actually, if you look across different brain regions, they are affected to varying degrees. So certain parts of the brain seem relatively refractory to the disease process, such as the cerebellum and the thalamus, whereas other parts of the brain, as I say, the basal ganglia, the striatum, take more of a hit. But as you can see, there are other areas, including the hippocampus, which I'll come back to, as well as the cortex. And this is very important because actually being able to image the brain, which we can now do with great skill using MRI and with great accuracy, allows us to start using MRI measures of shrinkage of the brain, volume losses, as a marker not only of disease progression in people who have developed the disease, but also as a way of actually picking up the disease before it's become clinically manifest, because shrinkage or cell loss and dysfunction in the brain, as I say, precedes the clinical features. And so a very important study led by Sarah Tabrizi in London called Track HD did just that, looking at people with pre-manifest disease, i.e. before they have clinical features, and manifest disease patients who clearly have abnormalities in keeping with the condition, and showed that there are fairly linear changes across certain structures, particularly the striatum, which may be useful as a way of predicting when the disease processes have started, as opposed to when the disease is expressed, as well as being a useful way by which to measure how people are progressing, against which we could try disease modifying therapies as and when they are developed, which is something I shall come back to at the end of this talk. So, in clinic, as I say, what does this actually look like? So, as I've already explained, there is a disease process which starts, which is when the abnormal gene, which we've already mentioned, starts to impact upon the cells itself. So, at that point, we have cellular dysfunction within the brain, which leads to cell death. But it is some years later, as I say, could be 5, 10, may even be 20 years before we actually see the earliest features of Huntington's disease, which I'll come on to. And so there's been a great deal of interest in this ability to pick up the very earliest part of the disease process, because if we can, and we have disease modifying therapies, it obviously makes sense to use those at a point before the patient has advanced to having relatively advanced disease pathologically and starting to develop clinical features. The question is, what are reliable measures which allow us to predict whether patients are in the very early stages of the disease process or about to become manifest? And that has been an area of great interest. As I say, imaging has been used, and we've been part of another study called Predict HD, which tried to ascertain whether there were certain subtle motor and cognitive measures which may be predictive of when people are about to develop the disease itself. And there are some subtle abnormalities which may be helpful in that respect, around changes in their motor performance, as well as some of the tasks which target the front of the brain, namely the executive aspects. So things like remembering numbers, being able to sort out problems online. So these type of deficits may be the very earliest feature. That, coupled to the abnormalities we see on early MRI scans, may ultimately be where we go with this condition, to say that the disease has actually begun in an individual, even though they themselves are not aware of any particular problems. As I say, once the condition has manifested itself, it then progresses over a 15 to 20-year period, with most patients coming to the disease typically with an advanced dementia, an advanced motor state with relatively little movements that they can control themselves, plus or minus psychiatric problems, often with severe weight loss. So we've understood with Huntington's disease, this is an inherited disorder. We've understood that once the disease process begins, we might be able to pick it up with imaging as well as subtle clinical measures. But there comes a point at which the patient clearly has manifest disease. In other words, there's a point at which someone can observe the patient and see that things are not right. And the question is, well, what are those abnormalities? What are the earliest features of Huntington's disease, and how do they progress over time? And here are a number of measures which I think are quite useful as indicators that someone has the earliest features of the disease. The first of these motor abnormalities, where people start to develop problems fidgeting, typically in bed at night, often not noticed by the patient themselves, but noticed by their partner or spouse. So these, I think, are very early features, and they represent the choreic dance-like movements that you see and which are well known to be associated with this condition. Cognitively, the earliest features are often patients having difficulty making trivial decisions. What do I order in a restaurant? What do I decide to buy at the shop in terms of getting this packet of biscuits versus that? Decisions which in themselves are not that important, but when they're low valence, in other words, they don't really matter, they often have great problems with this. So these are the early features: movements in the legs at night, difficulty making trivial decisions, and that's often coupled to psychiatric problems where the patients are a little more irritable than they used to be. They get a bit more anxious. They have mood swings where they can get very worked up over minor things very quickly. And generally speaking, the person who knows them best thinks there's been a change in the personality and a change in the way that the person is behaving. And this links into some problems of social cognition, the ability to be able to relate to others and understand what others are thinking, particularly around empathy. And sometimes this is also associated with weight loss and apathy. Now, obviously, these features are not specific for Huntington's disease. One sees a lot of people as they get a bit older who perhaps fidget a bit, have difficulty making decisions, get anxious, get more irritable. So, in themselves, you can't diagnose somebody, but in the right clinical context, namely someone who is known to carry a gene for Huntington's disease, this constellation of problems would make one highly likely that they are actually in the early stages of disease. It's also more complicated than that in that patients, by virtue of the fact the disease targets the front of the brain, often have a lack of insight. So we will have patients presenting to clinic who have absolutely classic features of relatively advanced Huntington's disease who will actually feel that they have no problems whatsoever. So you can have situations where people have manifest disease but don't actually complain or have any symptoms of Huntington's disease. Conversely, you can see people who carry the gene who are very aware of the fact that they are at risk of getting the condition, who then become very introspective and everything gains a prominence which it probably doesn't merit. So they think that they have Huntington's disease. So these are people who are polysymptomatic, but for which you have no physical features indicative of the disease. So it can be extremely difficult to know whether someone is in the early stages of Huntington's disease or not based on what they tell you and what you find when you look at them. This is why we prefer the term pre-manifest and manifest, because symptoms rely on what the patient are telling you. And as I say, you can have people with manifest disease who are asymptomatic, and you can have people who don't appear to have Huntington's disease who are polysymptomatic. So, how do we best serve our patients with Huntington's disease? So, how do we manage our clinic here in Cambridge, which in some ways is a useful way to think about how you could manage this wherever you're looking after these patients? So, the way in which we manage our clinic is that we run a service where we have patients who come up to a specialized clinic, which contains a neurologist, which is myself. We have a neuropsychologist who obviously goes through the cognitive tests. In the United Kingdom, we have regional Huntington's Disease Association workers who liaise between our clinic and the community with the patients, which gives you an insight into how things are for them and also takes information out to the patients themselves. Linked to us, we have a neuropsychiatrist, given the psychiatric problems are common in Huntington's disease. And importantly, we have a clinical geneticist who actually will ensure that we go through the right processes for predictive testing, which is where the patient comes from a family, which is to know whether they carry the gene but have no features suggestive of the condition, as opposed to diagnostic tests where patients have clearly features which would be in keeping with manifest disease, and the test is being done to confirm what you suspect clinically. Each of this needs careful counseling because obviously the consequences of having a positive test doesn't finish with the patient, it has ramifications for the whole family. And so in our clinic, we see patients who are from pre manifest, as I say, pre manifest gene carriers to those who have manifest and advanced disease. And obviously, we work with the community services which support these people in their home, including, as I've already mentioned, the Regional Huntington's Disease Association worker, speech and language therapists, social workers, physios, dietitians, and such like. It's always important, and this is something which I have emphasized to some of my colleagues locally, is that if you're seeing someone who may have Huntington's disease, but this has never been broached at all with the family, or there's no family history, which can occur in some cases, then it is very important to make sure that the patient isn't referred directly to a Huntington's disease specialist clinic, or if they are, that the appropriate information has already been shared with the patient and their family, given, as I said, the ramifications of it. So my own practice is if someone is thought to have Huntington's disease, but that has not been obviously discussed with them or the family, then I see them in a general neurology clinic before referring them on to my Huntington's disease specialist clinic. So now let's look at these in a bit more detail. So what does this actually look like clinically in terms of some of these early features? So as I said, the motor features are very much these fidgety movements in bed, which are noticed by the partner or the spouse. Now, the way in which we actually first noticed this was a number of years ago when we actually embarked on a study where we were trying to capture more objectively the choreic movements of Huntington's disease. So this was nearly 20 years ago when we used a device called an ActiWatch. So this, can you believe it was in the days before Fitbits and all of these Apple Watches. So patients wear essentially a wristwatch which measures activity. And in this slide, we can clearly see that, as you would expect in people who have no Huntington's disease, so you or me, during the day we're very active, which you can see by all these black bars of activity. And then at night when we go to bed, we are largely inactive. So there's a very clear division between activity and non activity with our waking day and our nighttime activity. Now, if you look at people with Huntington's disease, which of course is what we were doing to pick up this objective measure of career, whilst we couldn't pick up career because career on an ActiWatch is no different to any other movement, what one could see is that this division or this clear distinction between night and day blurred so that patients' movements actually during the night were not absent and were more prominent. And in some ways, their activity during the day was less. And this, of course, fitted in with what we discovered clinically when we asked patients about movements at night in the early stages. But it also suggested something more important, which I'll come back to a bit later, which was that if patients were moving at night, typically if you have a centrally driven movement disorder, such as the career of Huntington's disease, the tremor of Parkinson's disease, when you go to sleep, that stops. So if these movements were occurring at night, it implied that the patients were sleeping in the way that they should do. And so we subsequently went on to look at this in terms of actually formal polysomnography, bringing people into sleep clinics. And we found that actually one of the early features of Huntington's disease, and as I say, I'll come back to this, was that the sleep architecture and their ability to sleep normally becomes abnormal very early on, which may be why these movements are more prominent at night. So, ways to pick this up in the clinic are that you can obviously ask for the movements at night. But the other way to do it is to get patients to do various activities and watch carefully to see if there are subtle abnormalities. So, one test we do quite a lot is we get the patient to repetitively tap between two targets about half a meter apart. And one of the things you can pick up is the way in which the movement is not quite regular. So, early irregular hand tapping can be an early feature of career, which you might not be able to see overtly in the clinic. The other way to do it is to get patients to do other activities. So, if patients are sitting talking, or if you get them to walk up and down the room, you will often see the little flick of the shoulder, the fingers, the face, and such like as they're moving along. So, often distracting the patient or getting them to do some other motor task brings out these movements. It is also important to know that not everyone has very overt or obvious career in Huntington's disease. So, patients with very early onset disease, so called juvenile Huntington's disease, who present under the age of 21, tend to present with almost no career, but a lot of stiffness, slowness, look very Parkinsonian with myoclonic jerks, possibly a tremor. And many patients, as they advance through the condition, in the more advanced stages, become very slow, very stiff, dystonic, without much in the way of career. Now, the other things that are worth looking for early on in a patient with Huntington's disease to try and help confirm they've moved into the manifest stage is to look at their eye movements. So, saccadic eye movements are often abnormal. Patients have difficulty initiating them. There's a thing called the Luria tri-step test where you get the patient to do three separate motor actions, one after the other in a sequence. And often patients struggle to put that sequence together. And the walking can often be abnormal in a rather difficult to define way, but clearly things don't look quite right. There's a little bit of cerebellar ataxia, there's a little bit of involuntary movement, there's a bit of slowness, there's a bit of clumsiness with it. But often getting patients to walk will reveal that it's not quite normal. And those subtle abnormalities, namely a problem with the walking, difficulty with the motor sequencing, slightly abnormal eye movements, and these subtle choreic movements, often alert one to the fact that the patient now has definite manifest motor Huntington's disease. Now, cognitive tests, as I've already said, often can be asked for around making trivial decisions, as well as asking them how their general thinking and ability to solve problems has been affected of late. That is a little bit difficult because depending on your mood, depending on your job, determines how obvious and how bad those may be. In the clinic, we tend to use a number of tests. These are very standard cognitive tests. One is called the Stroop test, and the other is the simple digit test. The Stroop test is a task where you get people to either name colored bars, read the word which is describing a color, and then you have an interference where actually the color and the word are actually different. And people with Huntington's disease in the very early stages find especially the interference task rather difficult. There's also a simple digit task where you're asking people simply to take symbols and convert them into numbers. And again, patients' ability to do that quickly and efficiently can be affected early on in the disease process. So these are useful tests by which to judge people as to whether they may have early features of the condition, which becomes increasingly important in some people who have jobs of great responsibility. So air traffic controllers, doctors, people in professional careers, where the ability to pick up these very early high-level cognitive tasks will have major impacts on their career. So you have to be certain that they actually have them. But similarly, not addressing them means that they may be making decisions which affect others as a result of the disease process. It's also been said that everyone with Huntington's disease will develop cognitive problems of some sort, and I think that is true. It's also said that everyone with Huntington's disease will eventually end up with a dementia, and that I'm less convinced about. So we actually did a study a number of years ago using a test called the Edinburgh Cognitive Examination, and we looked at patients at various stages of the disease, both early, pre-manifest, as well as advanced disease. And what we found that whilst there is clearly a trend for people to develop early cognitive problems, particularly of the frontal executive type, which progress into a more global dementia as the disease progresses, not everybody follows that path. So, for example, a quarter of our patients in late stage disease appear not to have a dementia as judged by scores on these various different tasks that we use. And similarly, about a quarter of patients at the point of diagnosis, so these are people who have very early subtle motor abnormalities, actually have severe cognitive problems to the extent that you would say they have a dementia. So the rate and time at which people develop cognitive problems does vary with this disease, which highlights just how heterogeneous it is, despite the fact it having a single genetic cause. Furthermore, there's classic abnormalities in Huntington's disease, which are said to be these frontal executive problems, aren't also the only problems that people have in the early stage disease. So, a number of years ago, we were interested in looking at the hippocampus. This is the area of the brain that's very much involved with spatial navigation. So, the most famous study on this is the one done in London, showing that London taxi drivers have a particularly big hippocampus because they presumably spend a lot of time navigating around the city. So there are tasks where you can look specifically at this type of memory in patients, in animals as well. There's a task called the Morris Water Maze, which simplistically is you put a mouse or a rat into a tank full of water in which there is milk so they cannot see through the water. There's a hidden platform, and around the swimming pool, there are a number of objects which they use to orientate themselves to the platform. So when the mouse goes in initially, it swims around, doesn't know where it's going, looks around the side of the pool, and eventually it finds a platform, and it will work out that the platform is between, say, the clock and the window. The next time you put it in that tank, it will say, I know the platform is between the clock and the window, so it swims much more directly to it. So we tried to set up a similar task in patients using a virtual Morris water maze, because obviously we couldn't put people in a swimming pool with their swimming trunks. And what we found was that actually in patients early on in their condition, they have major problems with this. There is a more subtle way of looking at this particular type of problem called the paired associates learning, the details of which are not important. But the important message is that actually patients in the pre manifest stage, i.e., before they've developed overt motor features, have deficits in hippocampal based tasks, which may be a very sensitive measure by which to say that the disease process has begun. So, coming back to a point I was making earlier, this type of test may be useful in the future as a way of being more reliably able to tell a patient the disease process has begun, and they may therefore be suitable for a disease modifying therapy when and if such trials come along. The next area where patients often have early problems which can dominate the disease, again, it varies greatly from patient to patient, are psychiatric problems. The classic ones, as I've said, are irritability, so people getting worked up about things, not really being able to take things in rationally. They get anxious, they get very apathetic, and they have major mood swings. Now, these can be so major in some people that they can actually end up coming into the criminal services. So, I've had various patients who have presented through the prison services where some criminal act has been done. And because it wasn't particularly known that they had Huntington's disease, or in fact, this was the first manifestation of it, they can end up being incarcerated, put into normal prisons without any consideration being given as to what may have driven, in many of these patients, some abnormal behavioral problems. So, it's always worth bearing in mind that if you're referred people from such services, to think carefully about whether they have Huntington's disease, because obviously, whilst it doesn't allow them to get away with the crime, it is mitigating circumstances. The other area which I think has probably been slightly overlooked but has become more prominent in recent years is this higher level cognitive function. So we've talked about the early motor features, we've talked about these cognitive problems around executive functions, around hippocampal memory type functions, we talked about the psychiatric manifestations. There are also problems with social cognition and a lack of empathy. So, this is very much theory of mind. So, this is the type of cognition where if you look at somebody, you can see that they are upset, you can see that they're cross, you can understand their emotions around a certain action that either you're doing or there's some independent parties doing. So, this ability to be able to read people's emotions is obviously very important for everyday life, it's very important for employment. And so, if one did have a problem in this area, it could have major implications in how you actually conduct yourself and do your work, which may be easily overlooked, but may also explain why many patients with Huntington's disease often end up in trouble at work, not just because of the straightforward cognitive deficits and the psychiatric problems, but because of this inability to be able to read the emotions and such like of others. So we've looked at this over a number of years. My colleague Sarah Mason has done this. And again, the details aren't important, but what we found was if you use this task of reading the mind in the eyes, which is where you, for example, have a test where you get people to look at various faces, they have to imagine what emotion is being portrayed in the eyes of that patient. What we found is that patients, when they have manifest Huntington's disease, are extremely bad at this task, and those who are in the early stages or at the stages just before they become manifest also have major impairments in this. So it is clear that actually these higher cognitive functions, these social cognition tasks, are affected early on, which we've yet to explore the extent to which they impact on their employment as well as their ability to relate to others. So coming back to the point I mentioned earlier around the ActiWatches is this idea that there are more systemic problems in the body. So they have weight loss, which can be a major feature, and poor sleep, which I've already intimated by the fact that you could see these movements at night. So this was something which we were very interested in based on that early work that we did with ActiWatch. So we thought we would explore this further to see the extent to which this is actually really true of pre-manifest patients, the extent to which it relates to when patients get the disease, and to some extent, what is the neurobiological basis for this? Is this related to the Huntington's disease affecting critical parts of the brain? So we embarked on a study a number of years ago where we collected patients with pre-manifest Huntington's disease. So as I say, people with absolutely no features of the condition at all, either cognitively or motorically. We collected a matched group of control patients. And then we undertook a number of studies where we put them in a metabolic lab and studied them over 1 to 2 days. We also studied them in the community over a month using particular measures to study their metabolic activity, as well as then doing at the same time sleep studies, both within the home environment using ActiWatches as well as in the sleep laboratory. And what we found in this study was that there were no major metabolic problems. So, although we had suspected based on earlier work that patients with Huntington's disease are very catabolic, i.e., they have an increased metabolic rate, so they're always breaking things down, hence their weight loss, we didn't find this in pre manifest patients. We did find some problems with bone mass and less energy expenditure, which we couldn't quite explain. So, there may be subtle metabolic problems, but there was nothing majorly wrong in these patients. However, what we did discover was that there was abnormalities of sleep which preceded the development of the disease, both in terms of their motor performance and interestingly coincided with their early cognitive deficits. And what we found was that patients tended to go to bed earlier than their match controls. They got up slightly later, so they seemed to spend more time in bed, but their efficiency, i.e., how well they slept over that period of time, was less than with controls. And their ability to organize their sleep using different sleep phases, which we measured using EEG recordings from the brain, was very abnormal. So, this implies that one of the earliest abnormalities you see in Huntington's disease is the capacity to organize and regulate and hold your sleep in a normal pattern. Now, there has, of course, been great interest in what is the role of sleep, especially in the current age where we spend all our time flying and looking at electronic devices. And it's clear that sleep fulfills a very major part in making our brain healthy, both in terms of synaptic health, cognitive functions, and actually retaining the brain so that it doesn't actually degenerate, which seems to be a major problem in people who have huge sleep problems, in that degenerative diseases of the brain are much more common in such individuals. What we discovered was that not only do we find these abnormalities in early Huntington's disease, but actually it coincides with the cognitive deficits. Now, whether these happen independently, sleep falls apart and cognition falls apart, or whether the abnormal sleep drives cognition, we're currently in the process of trying to address, but it implies that some of the very early features of Huntington's disease may, in fact, be a reflection of chronic jet lag. So that is the sort of clinical features of Huntington's disease. I've described some of the motor features, the early fidgety movements, the Parkinsonism and dystonia you see in juvenile and in late stage Huntington's disease. I've described the range of cognitive deficits, both in terms of standard cognitive measures, executive frontal type tasks, hippocampal spatial memory type tasks, as well as these higher order social cognitive deficits and empathy. We've also discussed some of the psychiatric features and how they can be a prominent early feature, which may mask some of the other features and get people into trouble before they're actually seen by the medical profession. And then we have these new, more exploratory areas around where there's a fundamental problem with sleep and possibly metabolism. The next question is, well, how do you actually treat these patients? And so there are actually very few controlled trials of agents used in the treatment of Huntington's disease. There is a trial of tetrabenazine, which has been shown to actually deal with the career of Huntington's disease, which is used. Our preferred option, I have to say, is to use a drug called olanzapine, which is an atypical antipsychotic. And the reason we're very keen on that is that this is a drug that not only helps with the career of Huntington's disease, but it also can calm patients down, which helps with some of the psychiatric problems. It's helped people sleep, which, as I say, can be a problem. And it often causes them to put on weight, which can be a major problem in Huntington's disease. So this agent is extremely useful as a way of treating many features of the condition. But typically, the drugs we use to treat the chorea are olanzapine and then other neuroleptic drugs such as risperidone or sulpiride, as well as tetrabenazine. If patients present with the juvenile form, so they're more Parkinsonian and rigid, or the more advanced forms of Parkinson's disease where people are dystonic, we have very little actually which helps, but some people give them trials of L-DOPA and clonazepam. Cognitively, we have no agents which really help people with Huntington's disease unless the cognitive problems relate to psychiatric problems. So, if people are severely depressed, they can be cognitively impaired, which will improve when you treat their depression. In terms of treating their psychiatric aspects, there's nothing special about Huntington's disease. We tend to use the same agents as we would use in anyone else with depression. For psychotic behavior, which is actually, I think, relatively rare in Huntington's disease, we use the atypical neuroleptics. And for mood swings outside of olanzapine, I tend to use lamotrigine and other antiepileptics such as sodium valproate and carbamazepine. These other features, which I've mentioned already about the weight loss, we've discussed, but food supplements are very important. Sleep problems, we can use a number of different agents to help with that. And it's also very important to support the patient socially to make sure that they're linked into social services, that they're in appropriate housing, that they get the right level of care that's necessary in order to support them as best one can in the community. And this brings me on to the sort of other issues which are always worth bearing in mind outside of the pharmacological treatment of patients. So I would very much advocate that patients should be seen at a specialist clinic where the physicians and those attached to it know most about this condition and therefore are best placed to help recognize and support patients. There are clearly implications around driving and employment once you have manifest Huntington's disease in terms of whether you should still be allowed to drive or carry on with your job. It's important to make sure that their swallowing and their weight, as I say, is well maintained. So that involves speech therapy as well as their physical mobility through physiotherapy, and that their house is adapted to the most appropriate level for their disability, so occupational therapy. And obviously, looking after someone, especially with relatively advanced Huntington's disease, puts a great burden on the carer and the family. So, thinking about respite care, looking for specialist centres, and even thinking about nursing home placements is something that's to consider earlier rather than later. It's also important, I think, for the family and for those involved around the patient to think about power of attorney, advance directives, and also seeking to make sure that they get the benefits which they're entitled to through the welfare state. It's also extremely difficult with this condition in terms of some of the ethical issues that can arise. So, we have had patients who are gene positive who fulfill professional jobs within the military, for example. At what point are we at liberty to tell their employers that we think they've got the disease if they don't wish us to tell them? So, this can create issues. And also, there are problems around other family members knowing whether the patient you're seeing has it, and that can create tensions which can make it quite difficult. So, the final part of this talk is about disease modifying therapies which are being considered. And I'm not going to spend a huge amount of time on this because there is a large amount of work that's going on in this area, but I just want to flag up some of the interesting approaches that people are taking. So, probably the area that most people know most about at the moment is the idea that to target the problem in Huntington's disease, the most logical place to go is to either remove the gene or to remove the gene product which that abnormal gene is coding for, namely the mutant Huntington. And so there are large numbers of approaches which people are using to try and target either the gene, the messenger RNA, which then leads to the production of the protein. Now, obviously, this is a challenge in terms of delivery because if you really want to cure someone of Huntington's disease, you would have to remove the gene or the mRNA or the protein from every single cell in the body. And you'd have to do that consistently throughout life without affecting the wild type Huntington, which is made by the non affected allele. But antisense oligonucleotides are now in the clinic, and a number of companies are trialing this to see whether we can actually reduce the levels of mutant Huntington and by so doing slow down or even stop the disease. And I'll come back to that at the end. There are also ways not only to try and treat and get to the core of the disease by stopping the production of the abnormal protein, but by giving agents which increase the metabolism or the breakdown of that particular protein. So there is particularly a process called autophagy within the cell where abnormal proteins and abnormal organelles are actually broken down and removed. So, can we increase the clearance of mutant Huntington? And of course, those two are not mutually exclusive because you could reduce production with an antisense therapy and increase clearance through a drug that upregulates autophagy. Now, obviously, once the abnormal protein has been coded for within the cell, then that interferes with a whole series of other aspects of the way the cell functions. And so, a third major area in which you could try and target the disease process in Huntington's disease is to go downstream of the gene and the protein. So, look at the consequences of that upon mitochondria and upon how neurons relate to each other and transmit between each other. So, people have targeted those sort of areas. And then finally, there are areas where people have thought not about trying to stop the disease process itself, but to try and repair that which has gone wrong. And that has taken two major areas. One is to actually replace the missing cells with transplants of cells. The other is to provide a growth factor for those cells which are predominantly affected, typically within the striatum, either in the forms of cells that secrete growth factors or in terms of actually directly infusing growth factors themselves. So all of these have over the last 20 years been the subject of various trials, some of which have actually produced negative results. None have yet been proven to actually definitely slow down the disease process. The area that perhaps has received the greatest interest in recent years is this trial, which was originally funded by a company called Ionis, but is now being run in a larger study by Roche, which uses this antisense oligonucleotide to target the RNA of Huntingtin. And so this paper, which was published in 2019 in the New England Journal of Medicine, had 44 patients. And essentially what this paper showed was that as you increase the dose of the antisense oligonucleotide, which had to be given into the fluid around the spine, so was delivered intrathecally, actually the larger the dose, the larger the drop in the Huntington in the fluid around the spine, suggesting that this agent did actually interact with the messenger RNA and reduce the levels of the protein, not just the mutant, but wild type, but importantly the mutant. And as such, based on animal models, that should slow down the disease process. We don't actually know whether that's the case because we've yet to see whether this intervention makes a clinical difference to the patient, but at least it's an important first step to show that this approach does actually target the protein as we thought. So, to conclude, Huntington's disease is a genetic disorder and we can test for it. As a consequence of its autosomal dominant inheritance, it is very important that when one makes a diagnosis of Huntington's disease, that the appropriate genetic support is put in place and processed, because the implications for a positive test don't stop with the patient but have ramifications across the whole family. The classic features of Huntington's disease, which have always been thought to be the choreic movements, the evolution to a dementia with psychiatric problems, whilst largely true, is much more complicated than that. Patients present in many different ways, so there's great heterogeneity, and the range of clinical features extends out of just the chorea into Parkinsonian and dystonic abnormalities. The cognitive problems don't just target the front of the brain, but they also have hippocampal problems as well as this social cognition. And there are also other problems which we have identified around metabolism and sleep. So this disease is much more complex and is much more varied than we once thought, which obviously throws up challenges when we come to think about using disease-modifying therapies in the clinic, because obviously we're not dealing with necessarily one population of patients who are going to all follow the same course. They may present and progress in very different ways, and trying to understand that is something that many groups are working on at the moment, and there are some interesting insights on DNA repair mechanisms which may be contributing to that. There are many treatments which are being pursued in Huntington's disease. We have an armory of drugs which we can use to help some of the symptoms, which are often very effective in certain aspects of the disease. But at the moment, we have no disease modifying therapies. But nevertheless, there are agents on the horizon which are now coming into clinical trial, which offer great potential for slowing down or possibly even stopping this disease if we can use it at the earliest stages, such as the antisense oligonucleotides. So the future for this condition is very exciting. I think the most important thing we need to remember is that trying to understand the best way to treat this condition is not the preserve of one person. It requires the support of all the family, all the carers, as well as all of the medical expertise which is needed to manage this complex neuropsychiatric condition.